Hello, my name is Chloe Ilagan, and today I'll be talking to you guys about the tuberculosis vaccine, also called the TB vaccine, and it is also marketed underneath the name Bacille Calmette Guerin BCG vaccine, which is named after the researchers that developed this vaccine. So to start off, tuberculosis is an infectious bacterial disease that attacks primarily the lungs. It can attack also other parts of the body. But as you can see, these green dots, this is where the eight countries have the highest tuberculosis cases, and they're not exactly in the US, and that just comes to why the BCG vaccine is not administered in the US, but I'll talk more about that later. But in 2020, 87% of new tuberculosis cases happened in the 30 high TB burden countries. And so 8 of them listed here accounted for more than 2/3 of the global total. So I thought that was pretty interesting. We have India, Indonesia, China, the Philippines, Pakistan, Nigeria, Bangladesh, and then Democratic Republic of the Congo. And that was recently added in the past year or two, just because it has grown to have more tuberculosis cases in it. So in the BCG vaccine, we have Mycobacterium bovis, and it's the bacterium that causes tuberculosis in cows and badgers. And it's different from the tuberculosis-causing bacterium, which is Mycobacterium tuberculosis. Though they are different, they do have similarities. They are strict aerobes. They are very small, slow generating. They have the shape of bacilli, and they can be stained by using different techniques. But for Mycobacterium bovis, it is a live, attenuated vaccine, so it's in its weakened form to where it can go inside a human body or animal and just not cause bacteria or not cause it to increase. And so this is a picture of Mycobacterium bovis, and the mechanism of attenuation is that there's a loss of secreted lytic function. So we know what lysis is, it's just the destruction of a cell, but we need that reduction of that secreted lytic function needed to invade into lung interstitial tissue for the vaccine to work. And the mechanism of the BCG vaccine, it induces alterations in the histone modification patterns of specific genes that have to deal with monocytes and NK cells. So we did learn in our past class lectures that monocytes, they digest microbes, they deal with dendritic cells and macrophages, and then also NK cells, they secrete granzyme and perforin in order to kill other cells. But to go into depth of the mechanism, the first step is that dendritic cells, neutrophils, and macrophages, they recognize the BCG that is inserted into the skin at that site. And then dendritic cells are activated, which then migrate to lymph nodes in order to activate these adaptive immune system cells. Next, the CD4 plus and CD8 plus T cells, which are very specific to Mycobacterium itself, they are activated, and then they secrete large amounts of interferon, gamma, and granzymes. Next, the B cells are then activated, and then we have the memory and plasma cells, which are generated, as well as the antigen-specific antibodies, they are produced. Lastly, we have the memory T and B cells residing in the lymph nodes for maybe like a secondary infection of tuberculosis. So that's when the vaccine works. So this diagram just illustrates, like we see the BCG needle going into the skin. Then we have the macrophages, neutrophils, dendritic cells right here. Then they are the DCs are activated going to the lymph node, and then we have the antigen presentation, the APCs, and the CD8, CD4. And this video is covering actually the plasma cells and memory cells. But yeah, that's the overall mechanism. So what is exactly in the BCG vaccine? So we have this Tice strain. It's used in this BCG vaccine preparation that was developed at the University of Illinois. That was taken from the original strain that was developed in Pasteur Institute in Lyle, and I'll talk about that later in the historical perspective. But the medium in which this Tice strain was grown in was made up of glycerin, asparagine, citric acid, potassium phosphate, magnesium sulfate, iron ammonium citrate, and then in the final preparation, there was lactose present. I don't know the main functions of each specific ingredient, but I know that they are all key to making a certain medium in order to cultivate this bacteria. And there are no preservatives involved. So looking at this table right here, we have the adjuvants used in recent tuberculosis vaccine candidates. I don't know what these mean, but I know they are like certain different lipids or microorganisms. And so the majority of current tuberculosis vaccine candidates contain adjuvants. However, we need a tuberculosis specific adjuvant because it can induce a stronger immune response in the lungs and in the body. And so we have here the first column, adjuvant, and then we have the antigen. So adjuvants are just substances that enhance the immune response to a certain antigen. So we have, like, for example, H56 or M72 or H1. And then we have the adjuvant target, which is self-explanatory because it just targets it. And we have TLR, so this obviously stands for toll-like receptors, which mediate between the innate and adaptive immune systems. And we have endocytosis, lysosomal disruption, those certain processes that the adjuvants target. And then the last column, we have immune responses. So we learned about Th1, Th2, so Th1 differentiation, and other processes like that. NK, meaning natural killer, we have B cells, cytotoxic lymphocyte, dendritic cells. So as we can see, these adjuvants can be put into the vaccine in order to enhance a stronger immune response. So the vaccination schedule, this is actually quite interesting. It's only recommended for children who have a negative tuberculin skin test, who cannot be separated from adults who are untreated for TB and have TB caused by strains resistant to isoniazid and rifampin, and these are just like medications that help with tuberculosis to kind of decrease it. And it's only recommended only for children who are a few days old or up to 6 months old. So if they forget to get the BCG vaccine before 6 months, they have up until 5 years old to where it can be the most effective. Surprisingly, this vaccine is not recommended for adults 16 years old and up, so adolescents and adults, only if they're at risk. So if they have like a negative tuberculin skin test or they're just exposed to tuberculosis in family, work, sports, etc. And that's because studies have said that it's just not effective in adults, and they just saw more effectiveness in children. So thus, boosters are needed in adults if they do end up getting one, and there are no boosters for kids underneath 5 because it's just so effective, like I believe 80%. And interestingly, it's not required by the Dallas Independent School District. However, it is required by UD only if you're at risk. And I'm going to grad school at University of Houston College of Optometry, so I actually had to give a negative tuberculosis test to them to show that I'm not at risk. So it just varies between each district, school, area. Now the route, it's given intravenously in the upper left arm, kind of in the deltoid area. It requires trained medical personnel, obviously, because no one can just insert this vaccine into someone. And it has hypodermic needles and syringes, and that's what's needed to administer it. The adverse effects, there's going to be a white lump that's going to occur after the injection, and there's going to be, as you can see in this picture, like a red spot, like small swelling after 2 to 4 weeks, and then it's going to be called like a pustule, and that's going to heal after like 2 to 3 months after that vaccination. And the main side effects that will occur are soreness, the discharge at injection, fever, swollen glands underneath the armpits, headache, and that all has to do with like the localized inflammatory response, like the swelling and the pain and the redness. Another way that it can be administered is percutaneously, which means passing through the skin. So it's like not by like a needle, but just this multiple puncture device. So you're just gonna like position your arm horizontally, and then the nurse or doctor will apply 0.2 to 0.3 milliliters dose onto that clean skin surface. And then what that person's holding is a multiple puncture device. It's gonna go over that vaccine. So the vaccine is like that gray circle area right here, and you're just gonna apply pressure for about like 5 seconds, and just to make sure that the vaccine is applied enough. And then after that, just release, stay there, and it's going to dry. You have to let it dry for at least, I think, 24 hours. And so that's another way that you can apply this BCG vaccine, which is pretty interesting. Okay, I know this is a lot, but this is the historical perspective on the timeline, because I found this the most fascinating when I was researching about the BCG vaccine, just because World War I and World War II were happening. So I want you to keep that in mind. So we have the discovery of tuberculosis in 1882. Then Calmet and Guerin began their research in 1900s. But then there was an outbreak of World War I, and that kind of interrupted their first vaccination trial in cattle. But then we got to find the first successful vaccine, which was administered to animals. That's when we named the vaccine in 1919. Then human administration was performed in 1921. That's the current vaccine that we get today. So actually, it was administered to an infant who was just born from a woman that actually died a few days after giving birth. And that infant received it orally, so the oral route. And then 114,000 infants were vaccinated without complications via oral route from 1924 for about 4 years. And then came the Lubeck disaster, which is crucial because it was kind of like an outbreak. There was an accidental contamination of the vaccine. 73 infants died, 135 were infected, but they recovered out of the sum total 250 vaccinated. And then around that time, there was the intradermal route that was discovered. And I wanted to point out that Selman Waksman, his discovery of bacteriostatic activity towards MTB was just another effect of Calmet and Guerin's research. And then 1979, we have a Chinkoput trial, which their results were given to the public, but they just showed that they tested 2 strains and they just failed to prevent tuberculosis. And then 2015, the most common strains that we use today of this vaccine are BCG China strain and BCG Pasture 1173P2. So that is the timeline. And then lastly, just some of the interesting things that I learned was that the tubercle bacilli was cultivated on a glycerin and potato medium. And I just thought it was funny how they had to use potatoes, because this type of medium made it easier to suspend the bacilli. And then when they added ox bile, the virulence of the bacteria was reduced. And so that's what was needed in order to get that function of the vaccine going. And then in the beginning 1900s, the bovine strain of tuberculous was taken from the udder of a tuberculous cow. I'm still researching more about that, but I think that's where like more of the bacteria kind of grew, something having to do with their production of milk as well. But I mean, we start off with animals, and these animals are like good models. And because it was in the World War, potato costs increased because many prices of food were increased due to the war and all the trials. And it was difficult to obtain ox bile from the abattoir, which was like the slaughterhouse. So they did experience a bunch of trials and tribulations trying to develop this vaccine. Like, historical perspective, super, super important, because all the resources that went into it, the work, all that just made it unique to what it is today. And then guinea pigs, rabbits, cattle, and horses are the main animals that were involved with this discovery of the BCG vaccine. But in all, thank you for listening. This is all about the BCG vaccine. Thank you so much.